Once control is established, researchers can begin to separate two things that are usually inseparable: the stimulus and the perception.
The fleeting nature of smells is a problem for scientists. It's one of the central challenges to olfactory research, because smell is inherently unstable. Capturing a smell isn't the same as capturing an image or recording a video or audio file. You can't press play on a smell. Odor molecules drift, they mix with the air, they degrade over time. In testing smell, even small changes in concentration can have drastic effects on perception, which means that without precise control, you can't be sure what someone is actually smelling. So, if smell is so ephemeral, how do we study it scientifically? How do we isolate it? How do we measure it? How do we control it? Because if you can't control an odor, you can't measure it. And if you can't measure an odor, then you can't be sure what stimulus you're presenting. That is, if you can't control the stimulus, you can't trust the results. The perception of smell is triggered when an odor molecule binds to a receptor in the nose, sending a signal to the brain.
We have these physical molecules coming into our nose and matching up with our olfactory receptors and triggering a sensation. That is essentially the physiological system, the hardware that allows us to smell. And everyone is wired a little bit differently. But we're also probably even more differently wired at the software, the cognition, what we interpret those smells coming in. And that's interpreted based on our experiences. And those experiences could go all the way back to infancy in some cases, right? And so we have two different sources of input and two different sources of variation. What we can smell and how that molecule is represented in the brain, and then how the brain takes that information and interprets it. Is it good? Is it bad? Is it something I want to continue to smell? Do I want to eat it? Do I want to put it on my body? Do I want to get it away from me? Yeah.
An odor molecule, or odorant, is just a small chemical compound that can evaporate into the air and enter the nose. Once in the nose, it binds to a specialized receptor and initiates a neural signal that the brain interprets as smell. Not all molecules can do this. An odorant must be sufficiently volatile to reach the nasal cavity, soluble enough to interact with the mucus lining, and have a molecular structure that fits and activates specific olfactory receptors. An odor's ability to trigger perception is influenced by several factors. The concentration, because higher concentrations generally produce stronger perceived intensity. Also, the stability and delivery of that concentration, and the specific receptor interactions it produces in the brain. The brain shapes perception, learning, memory, prior experience. These are all influencing how odor signals are interpreted. Meaning that smell is not just a property of a molecule, but a constructed sensory experience. But for now, let's stick to the plan and just talk about the odor stimulus. For over a century, scientists have been attempting to measure smell with widely different results. I mean, they're looking at the same molecule, they're asking the same question, but it's different labs using different techniques and totally different results sometimes. Why? Because smells don't behave. They won't sit still. They drift and they stick and they dissolve. They change depending on the container, the air, even the room that you're testing in. So how do you study something that doesn't stay still? At the Monell Center, new research is showing that the answer might be a bag. Not a high-tech machine, not $1 million instrument, a bag. Researchers in Joel Mainland's lab have developed a new odor delivery system using something called a nalifan sampling bag.
So these are basically bags filled, so if you take a sniff of a jar, you draw room air in when you smell it, and so it dilutes it, but if you fill the bag with a known amount, and then you suck through a mask, and you get exactly the odor that you want.
Traditionally, using liquid dilutions of odorants is a standard way of preparing odor stimuli. These liquid dilutions are placed in bottles or jars or added to delivery systems that are meant to be used several times across many participants. But these methods lack the real-world experience of smelling odors as they travel in the air naturally. Also, with each trial, there's a concentration shift such that the stimulus is degrading a tiny bit each trial, so that concentrations may be quite different between the first participant tested and the 20th participant tested on the same dilution series. Instead of dissolving odors in liquid, the mainland lab lets them exist in air inside a sealed space, a sealed pocket of odor, so to speak. Using specialized sampling bags and sealed environments, the mainland lab can create precise concentrations of odor molecules and deliver them consistently. No contamination, no dilution, no variability from the surrounding air. Inside the sealed bags, the odor remains remarkably stable. In testing, the bags produced stronger signals, clearer differences between smells, and, most importantly, reliability. This might sound like a technical detail, but consistency of a stimulus is foundational to research. And smell begins with chemistry, because before you can understand how the brain interprets smells, you have to know exactly what the brain is receiving. Once that control is established, researchers can begin to separate two things that are usually inseparable: the stimulus and the perception. Not just what are we smelling, but how does the brain turn that into experience?